my presentation today is really very big picture, very general, not about the specific details of future CIAT, which is something I think we can talk about next year perhaps. I'm from Austria, I'm from Vienna. I grew up with these kind of foods, and I came to love Apfelstrudel and Sachertorte and chocolate cake. I used to eat like 1 kilo of chocolate bars in a single go. It was really addictive. One of the most special things I sometimes got for breakfast is this. It's bread with butter and a layer of white sugar on top. And that, at the time, I really loved it. I remember when I was a kid and I was asking for it constantly. At that time, that was the recommendation of doctors. Charge your kids with energy before they go to school. This may sound hilarious today, but are we really doing much better today? I really doubt. We're getting these kind of things today, which is just repackaged processed carbohydrates, sugar in more creative forms and colors. While at the same time, there is increasing evidence in the medical area that processed carbohydrates and sugars not only affect our health just by providing an excessive amount of calories, but also have all sorts of secondary effects that we have not really been very conscious about. For example, there's a lot of literature coming out over the last several years that cereal flour, very processed carbohydrates, sugar, cause profound changes in our microbiome, the collection of bacteria that live in our guts, to a point where the diversity of this bacteria is decreased substantially, more or less in alignment with sort of diversity of the food we intake in a way, I think. This can have quite widespread consequences. For example, there's an experiment where they had a mouse growing under sterile conditions. They took the microbiome of a slim and an obese mouse and transplanted that to a few sterile mice. And as it happens, the mouse that got the microbiome from the slim mouse continued to be slim, and the mouse that got the microbiome from the obese mouse suddenly turned obese, while both mice were eating exactly the same food. So even the microbiome itself can sort of reinforce obesity. Another issue that is coming more and more to the fore in medical research is that certain types of foods and the microbiome that feeds on it seem to be causing what is called intestinal permeability, which leads to the penetration of fragments of foods or bacteria into the bloodstream and causes all sorts of inflammation response and even autoimmune reactions. So all this research is going on, but pretty much in separation, I believe, from agricultural research. So perhaps it's no wonder that we are running the risk of trading one problem for another. The problem of food security against or versus the problem of creating increasingly obese and or populations suffering from chronic diseases. So I think we need more crosstalk between medical research, cutting-edge medical research, and priority setting in agricultural research. And I think we need to sort of realign our diets with what our physiology has been evolving to digest best over the last 1 to 2 million years. If we look today what we are consuming, we see that about 60% of all calories are derived from just 3 cereal grains. This is a very, very small amount of, or a small fraction of actually the whole number of crops that are being used globally for agriculture, about 200. And only in these 200 are really a small slice of the about 50,000 or so plant species that are edible, which again are just a small fraction of the about 300,000 plant species that live on our globe. Now, I think we should perhaps start thinking about that we not only need a Harvest Plus project to biofortify our diets, but also perhaps a Harvest Plus project to biodiversify our diets. We have the tools, the raw material in hands. It's the genetic diversity that's being conserved in our gene banks. But so far, we haven't really been able to use this diversity very efficiently because we have been focusing, very rightly so, on collecting genetic resources, we have huge collections, but we don't have much information about each of these accessions that we have collected. And this is where FutureSeed comes in. FutureSeed is designed to be a gene bank that continues to conserve crop diversity that has been collected, in our case in CIAT, through the decade-long work of Daniel in our gene bank, particularly in the case of beans, but also add to it a new kind of dimension, which is all about information about these genetic resources that have been collected and are being conserved. Basically, to help us to diversify our diets, but also to address these two other critical points, I think, that are the main areas where crop diversity is going to make potentially a huge impact over the next several decades, which is keep the environmental footprint of global agriculture in check, to not exceed the 23%, I think you mentioned, Christian, of terrestrial surface that is still in native form, as we need to increase production to keep up with population growth, but also, of course, to adapt agriculture to climate change. So FutureSeed is, of course, going to continue to conserve the collections we have, but also add this information, this dimension of investigating the genetic potential of these collections that are being conserved by adding information about basically what are these individual accessions good for in terms of application in breeding programs. But we also have to consider that we are basically right in the middle of a global hotspot of biodiversity. We are in Colombia, it's one of the most biodiverse countries. And there are several gene banks in Colombia and other countries in the regions that hold very, very unique crop diversity of other crops that are potentially very important in terms of diversifying our diet. Now, at the moment, the sort of political or the policy framework of exchanging genetic resources is a bit difficult, because it has become very difficult to exchange materials using all these different material transfer agreements, question whether or not crops are within or outside the treaty. But so we cannot necessarily easily exchange materials or perhaps offer space to conserve different kind of species, at least at this point in time. But what we can do, I think, is we can form a sort of regional community of practice to work together to better mine genetic resources that are being conserved, not only in our gene bank, but also in other gene banks. For example, Costa. And here's where the collaboration with Costa comes in, where we have our neighbors here in Costa who have a gene bank in Bogota, here as well. They are working on a range of different, very interesting species. And once we talk about applying genomics tools to mine these collections, you're dealing with DNA, you're dealing with information, it becomes very generic, so we can work together on a single platform. So what is really missing is information. So in a way, gene banks, to some degree, still resemble libraries without sufficiently informative catalogs. And what we cannot do really is evaluate each trait of potential interest for each of the varieties. What we need is a sort of generic technology to create a catalog of actionable information. The logical thing is to go to the sort of hidden causative layer, the DNA, the genome, that underpins all the phenotypic variation, and use information that we can gather there now quite cheaply, because sequencing costs have dropped about five orders of magnitude since the turn of the century, to use that information as a proxy to predict phenotypic traits across the whole collection. In some cases, it's quite easy to predict those traits, and the genetics is fairly straightforward, a single gene involved, but sometimes we have quite challenging relationships between, let's call it the genotype and the resulting phenotype. In those cases, I think we can work with colleagues from the big data platform, hopefully, to deploy deep learning, machine learning technologies to predict these traits for these for the accessions from the DNA level. The other thing is that we have not only learned to read the DNA hard drive, the genome hard drive, but we can also now write to it. And this is the technology of genome editing, which Joe has already outlined this morning. So we can, instead of having to do multiple crossing schemes to sort of assemble the optimum combination of different alleles in different loci. We can take one cultivar, eventually in the future, it's not yet quite possible, but I think we're getting there. We can take one cultivar which has already some combination of favorable alleles and then just override the unfavorable alleles with the favorable ones in the other locations. We can also, I think that there is already an ongoing discussion about using this genome editing technology to accelerate the domestication of new crops, which is outlined in this graph here. Essentially, the idea is here that many so-called domestication genes are already quite well characterized. Shattering genes or height genes. And the molecular changes required, or the molecular changes that differentiate wild from cultivated are well characterized. We can apply that knowledge to new species, to domesticate them, to bring them into agriculture much more quickly in the future. So all this depends on information. And that's the reason why we're adding a digital gene bank component to future seeds. That will be actually a physical building to hold what is called digital gene bank. It's one of 3 pillars of future seeds. The first one is, as I mentioned before, we're going to continue to conserve using the latest quality standards according to international standards, and also due to our relationship with the Crop Trust, we have to follow very strict standard operating procedures. This will continue, and we will continue to collect diversity if and where it is still missing, and we will be able to tell actually what is missing based on the molecular analysis that will be conducted. The second pillar I mentioned just will be the digital gene bank. The idea really here is to provide a platform for collaboration in the region on the topic of genomics of gene banks. And there are many questions that people come across when they start applying genomics to gene banks collection, which are quite common across different crops. So we would like to provide a meeting platform for CIAT, Corpo ICA, von Humboldt, other gene banks in the region. We are collaborating quite a lot with Costa Rica, for example, to come together and learn from each other while we are deploying genomics to gene banks. And the third component will be education. Collaborating with universities in the region and elsewhere to educate a new generation of scientists that are conversant both in genetic resources and also in these new molecular technologies. But also generate more public awareness here in Colombia about the importance of biodiversity, of crop diversity for agriculture and for the future of the planet. So I think it's a strategic investment. It's a strategic idea to be implemented in a strategic location at a very critical time. And I think we're very, very happy with the support that the President of Colombia announced yesterday, and Luis Castilla from the UK Embassy represents another contribution that will contribute to making this a reality. Thank you very much.
So we've seen a lot of exciting topics today and a lot of innovation. The Future Seeds project represents one of the really types of integrative efforts that a center like CIAT can do, again, in the context of something like the CGIAR. And it does show the value of having the umbrella type organization like the CGIAR as part of the respaldo, as we'd say here, for this type of effort. With that, I'd like to hand it over to Luis to sort of start the dialogue. We saw a number of different aspects. We've been hearing about the sort of the biophysical component, the agronomic component related to these crops, the diversity aspect, the genetic and computational aspect. But what we haven't heard a lot about is the social aspect and the social dynamics. How do you see something like Future Seeds fitting into this broader human context as well?
Yeah, that's definitely an interesting point. First of all, I would like to go back before I answer that to the point in which this caught the attention of the UK government. I represent a team who sits in Bogota, but constantly works with colleagues in the UK that is devoted to promote and work here on the science and innovation area to promote socio-economical development through partnerships between UK organizations and Colombian organizations like CIAT in this case. So when we initially heard about this idea, we obviously thought, okay, this can easily become a priority for us because it fit in our spectrum of work, basically because, as I was saying, we promote socioeconomical development, and we do it basically through two main tools. One main tool is the Newton Caldes Fund, which is contributing to the Future Seeds program. That's a UK government fund devoted to, and you might have heard about it already, to promote scientific collaboration and use science and innovation to promote development. And the other tool is the Global Challenges Fund, which is a newer fund. You might have already heard about it. There are some active groups already in Colombia working with it through the Erlami Institute, which is an institution, UK institution who won an award. And this is how we do it, I mean, through these two tools. And going back to that moment when the original meetings, I wasn't working for the embassy at that moment, but I know that our team held these meetings with the CIAT team, and that immediately triggered all the idea of contributing with this huge program, particularly because we are so interested as part of our science and innovation portfolio here in Colombia. Food security is one of our areas of interest, and that's how we decided to become involved and support this initiative. So it's a big honor for us to be working with CIAT and to be here celebrating the anniversary, sharing these good news. Definitely we are supporting this, and we hope this becomes a reality very, very soon. Conscious of time, I know, going back to your original question, I think, I mean, the social aspect of this, I mean, from the social sciences perspective, I think Peter was mentioning the three dimensions, and I think the public engagement one, the third one, is the key one for this, particularly because, yes, I mean, you're going to be working, I guess, with students at universities, with researchers at university, and so on. But there's also a huge component, and I will definitely, first of all, encourage it, and second, keep supporting it because it's so important. In our experience, and I know you have faced the same problem, communicating science is a difficult task, and talking to non-scientific audiences is always a challenge. And I think that the public engagement component of Future Seeds is going to be crucial for that, particularly because we need to be, again, working as partners, not as donors, as we heard in the morning, to be committed to making sure that people understand how Future Seeds and the whole science that is behind it actually affects their day-to-day lives, and how it changes through, in the end, having a healthier diet and a better quality of life. So I would definitely go for the public engagement component to make that happen.
Can I just add something to that?
Sí.
Gracias.
Go ahead. Little flip there.
That's fantastic news. I mean, just to give you an example, at the moment the genebank receives a little bit in excess of 1,000 visitors every year. And these, we're not advertising anything, we're just receiving a lot of interest from regional universities, schools, international, national visitors, and so on. And future seeds will actually have, I don't have the plans here, but there will be a sort of educational path that goes around the whole building. And the laboratories are designed to have open areas, like big windows, sort of aquarium kind of style of laboratories. So people can actually, from the outside, see in a sort of logical order in terms of workflow what is happening inside with explanations. What we would like to add there are some audiovisual tools to actually decrease the amount of time that staff is usually dedicating to receive visitors and lead them through the building. But these are kinds of activities that we cannot easily get funding for. Actually, it's really fantastic to hear from not you know the donor, you're an investment partner, or that there's interest in this area because now that we built the building, the infrastructure is ticked off. We can cover running costs for basic conservation activities through the genebank platform. We can use that to build on top of that to apply to seek additional funding for research projects in collaboration with breeding programs. But this educational component is otherwise very difficult to cover. So that's
and I'm sure it will come up. I mean, last week, I mean, for a different but still related reason, we had colleagues from different UK universities working with research councils in the UK, working with CIAT researchers as well on these topics. And I'm sure, I mean, whenever you mention, when you talk to researchers and scientists in the UK around how to communicate things, the whole public engagement arena starts action, and they are, and we are really focused on that at the moment. In general, in the UK, I mean, the whole science communication and the way you engage with the general public in the end is something we are really interested in. So.
And it's important to do that here in Colombia at this point in time, where suddenly a large part of the country opens up to be visited, to be explored, to be used for all sorts of purposes. Raising this public awareness about the issue of biodiversity, I think, is a very important objective. Yeah.
Well, and I mean, one of the things that we need to think about is where are the feedbacks, right? And this is a type of activity that if we do have wide public awareness and engagement across all levels, then it's going to start having a positive feedback and gaining momentum. And I think that Future Seeds is the type of nucleus around which we can create some of that positive feedback in order to actually create something that is sustainable over the long term.
And the beauty is also we are part of the science park. We're not only CIAT, and it will not only be CIAT who is working in this building, but Corpo ICA as well on joint projects and Corpo ICA projects. And Universidad Nacional is next door as well. So I think there are lots of opportunities for collaboration and getting that going.
Then likewise, the potential to serve as a basis for international collaboration, others coming here and participating in Colombia. Yeah. Well, let's thank our two presenters, and that'll bring this part of the CIAT 50 to a close.